Good morning and welcome to this conference on personalised medicine, the promise, the hype and the pitfalls. My name's Joshua Horden, one of the conveners of today's conference. I'm delighted that the Oxford Healthcare Values Partnership, which I lead, is collaborating with the Stratification in Colorectal Cancer Consortium on this project. Tim Maughan, who leads ESCORT, will be saying a few words about that shortly. A few words about the Healthcare Values Partnership. We're an Oxford-based organisation and we have a range of research projects that we work on. Team members in the partnership in a number of disciplines, including law, theology, palliative care, general medicine, general practice and commissioning. And projects which we work on include compassion in healthcare, thinking about marketisation processes, and this project on personalised medicine. And there's more about us on our website. We're very pleased to be holding today's conference in association with CASME Academic. That's the Centre for the Advancement in Sustainable Medical Innovation. And Professor Sir John Took will be speaking about CASME. And also the Centre for Personalised Medicine, Simon Leedham, who will be closing our conference, will say some words about CPN later. I want to thank in advance all those who've contributed to putting on today's conference, especially Dr. Therese Filer, who's the Wellcome Trust Postdoctoral Fellow with the Healthcare Values Partnership, and Dr. Kezia Gateskill, who's a pathology reg and doctoral candidate at the Department of Population Health. I'm also very grateful to Claire Garbus and Andy Blake from ESCORT. I want to say thank you to the chief funders of the conference today, and that's the Sir Halley Stewart Trust, represented today by Professor John Wyatt, and to the Wellcome Trust. Very grateful to them for making today's event free to attend. And thank you to you for setting aside time to participate today. We really want this to be an interactive and productive occasion, so please do feel engaged and able to contribute. Two bits of housekeeping. Toilets are outside and to the right. There are fire exits marked, but I think no drills are planned as far as I know. Please do come back to the sessions promptly if you would, so we can start on time. And if you do want CPD points for today's event, that will be available. You need to sign the list back at registration around lunchtime. That's in process. So, turn to our theme itself. As you may know, there's a significant critical literature relating to personalised medicine. And even in using the term personalised medicine, we're already into that critical enterprise. Should we really be saying stratified medicine? Because to use the word personalised is to suggest that medicine can be promised, which will be truly tailored and personalised to an individual person. But this may be less than plausible. Stratified may be a better option. It may be generally scientifically and clinically implausible that we can have this kind of tailored medicine, but also it may unkindly raise the expectations of patients and the general public. That'll be a major theme for today, as it was in our scoping workshop back in December when we started to lay out some of these questions. What is or should be meant by the term personalised medicine? That's bound up with questions of value of one sort or another. So there's a significant critical literature out there, and just to point to one example of that, this volume, The Ethics of Personalised Medicine, Critical Perspectives. So this was a collaboration led by Joachim Vollmann and funded by the German government in order to bring together a number of different specialists, voices from oncologists to theologians to policy makers. And two particular points come out of that project. First, the vagueness of the term personalised medicine, and then the question of values. And Vollmann suggests that there are three main positions which can be identified as to the meaning of personalised medicine. First, that personalised medicine is not a new concept, because medicine has always been individualised. Second, personalised medicine is holistic healthcare, centred on the needs of the individual patient. And third, that it's treatment targeted at stratified subgroups. And then they argue for one particular definition. They do an exhaustive search of 2,500 articles, 700 different definitions, believe it or not, of the term personalised medicine. And this is what they boil down to, that personalised medicine seeks to improve stratification and timing of healthcare by utilising biological information and biomarkers on the level of molecular disease pathways. So that's one way of defining the term. But perhaps it's not the first thing that people think about when they hear the term personalised medicine. There's a good number of voices concerned that there's a kind of mystique growing around personalised medicine, which generates unrealisable promises and expectations. And that emphasis on promise is going to be important for today. One of the things we're interested in is how clinical, commercial, and other sectors talk about personalised medicine. What promises are being made? How fulfillable are they? What drives the making of promises? And how is the potential of personalised medicine to be distinguished from overstatement, which amounts to hype? And how are we to assess whether hype, if it exists, is good or bad, or just perhaps necessary in order to keep the show on the road? So with this in mind, we want to consider the pitfalls which may lie in the way as we move down the road of applying stratification or personalisation in healthcare. 3 themes came forward in our workshop in December, which will focus us today. Details are there on the programme, so I won't go through it exhaustively. But they broadly cover personhood and genetics. Are we defined by our genome? How do risk and value relate to people's experience and sense of identity? Second, data sharing and participation, issues of perception, cooperation, law and education. And then questions of equity and justice in healthcare. How is all this affordable, and is personalised medicine just a rich world enterprise? Now, there's no point in controversy for controversy's sake. Our aim rather is to stimulate conversations which deal with controversies and for the sake of consensus. And overall with a shared aim of improved healthcare. And in particular, we're not interested in moral alarmism, which I think is a particular temptation for activist ethicists to see moral problems in every test tube. The challenge, rather, is to ask the right ethical questions in the right way and to find answers to them over time. Nonetheless, the field is controversial. There's vigorous, if polite, debate in journals such as The Lancet about whether the entire enterprise is ill-conceived and whether the focus of research spending should be shifted very substantially to public health. Some say that the promises are not going to be delivered, unlikely to be delivered in the future. Why might humanities disciplines such as theology be involved in debates like this? Well, we think that values lie at the very heart of healthcare in general and in personalised medicine in particular, defined in the sense which we've given just now. And humanities disciplines, I think, have a particular gift for critiquing, exploring the values which are in play in any given activity. Everybody has values, whether we're scientists, physicians, patients, policymakers, politicians, theologians. Everyone is invested in some way in an enterprise, whether professionally or as a patient. And humanities disciplines have the capacity to generate discussion about that investment and to press people towards thinking and asking questions about challenging values issues. Theology is one such discipline with that capacity, with its own diverse history, sources and mode of inquiry, capable of creatively reframing discussions and preserving what is on reflection good and illuminating what is on reflection not so good or not good at all. And it's not just us emphasising the importance of this values debate. House of Lords Science and Technology Committee report made similar observations on what it sees to be a generally troubled relation between science and society. Here's what they said. Some issues currently treated by decision makers as scientific issues, in fact, involve many other factors besides science. Frame the problem wrongly by excluding moral, social, ethical and other concerns invites hostility. Underlying people's attitudes to science are a variety of values, and bringing these into debate and reconciling them are challenges for the policymaker. So our aim today is to provoke discourse and debate, to identify aspirations which are more grounded in myth or hype than reality, and to challenge them, and to identify questions which need further examination. To enable this debate, we're going to ask all our speakers to be relatively brief, and we have an outstanding mix of delegates today, a wide range of academic disciplines, professions and policy areas, and we want to invite your comment and perspective on what we're discussing. As a way of fomenting that discussion, after this first set of talks, there'll be a couple of minutes to turn to those around you and to ask what you found interesting about the conversation so far. And when we get back to plenary discussion, We'd be grateful if you could identify yourself briefly so that we know who we're listening to and talking with. So, without further ado, I'm going to introduce our three speakers briefly for today, for this first session, and they're going to follow one after another, and then we'll have discussion at the end. So our first speaker is Professor Tim Maughan, and Tim is, as I mentioned earlier, one of the leaders in the S-Corp collaboration, Professor of Clinical Oncology here in Oxford. And then we'll ask Professor Sir John Took to speak, who's Professor of Medicine at UCL and co-chair of CASME, one of the groups sponsoring the event. And lastly, we'll turn to Steve Sturdy, Professor of Sociology of Medical Knowledge up at Edinburgh. So over to Tim first.
Thanks, Josh.
What you have to say.
Thank you. Great. Well, may I add my welcome to you all. It's great to have you here. Thank you for making the effort to come today. I'm an active researcher in stratified medicine, I would say. I'm a colorectal cancer specialist, a radiation clinical oncologist. I'm working here in the Institute of Radiation Oncology and have research in stratified medicine in treatment of bowel cancer and in trying to discover stratifiers. So I'm actively involved in this field. And there is enormous promise of personalised medicine. It's a very attractive concept for us as scientists, as clinicians, as patients, and the funders are right engaged in it. So many of you are engaged with the various MRC stratified medicine consortia. Here's the list. So colorectal cancers come lately to that field. As you can see, most of them are across a range of non-malignant diseases. And the MRC has said stratified medicine will ensure that the right patient gets the right treatment at the right time, and has invested quite a lot of money in it. I'm interested in that word, ensure. There's a big promise in that concept. This is going to deliver something very, very powerful. The EU extends it to include the right prevention and treatment for the right patient at the right time. This is big stuff. Not only are we going to be cured, we're going to be kept healthy all our lives. And this year, the big precision medicine initiative in the US, with very significant investment from the president himself. I'm very interested when you look at this work here, much more guarded and thoughtful comment on their literature than there is in some other places. So there's a big promise. And what is that promise? It's the promise that the treatment, which targets the causative molecular alteration, is specific for you and will be highly effective for you. That's the promise. And the great example of that is the use of a drug which we call Gleevec for the treatment of chronic myeloid leukemia. So this is a terrible disease which, when I was involved in treating some of these patients in collaboration with hematology back in the 1990s, we were doing non-matched donor transplants, and there was a horrendous procedure-related mortality of about 30%. And we were prepared to do that because the disease was so awful. But there's a fundamental molecular alteration here, and I know many of you will know about this, but because we're a very multidisciplinary audience, I'll be going over some stuff which isn't familiar to everybody. A particular gene up here, a new gene, the BCR-ABL fusion gene, which encodes a novel tyrosine kinase enzyme. Imatinib is a drug which specifically inhibits that enzyme. And CML is quite unusual. It's a single aberrant protein, this protein, result of this translocation. And if you go back to the phase 1 trial of this drug when it came in, very minimal side effects. 53 out of 54 patients achieved a complete hematological remission. What would we give for a phase 1 trial result like that? And here you can see blood counts going into the normal range. And this wasn't just a short-term effect. Event-free survival was 83% at 5 years. Previously, 1 in 3 patients only had survived 5 years before. This was a complete game changer and has continued to be so. Sadly, the reality has not been as good following up. So this paper a couple of years ago looked at the next 71 drugs approved by the FDA for cancer, personalised medicine drugs. The progression-free survival on average was just 2.5 months. You can see the ups and downs, and the overall survival benefit averaged 2.1 months. So this has been a bit of a challenge. An example of that, the BRAF gene, which encodes this protein in the signaling pathway, was one of the great finds of the Cancer Genome Project. And a drug, vemurafenib, was developed to treat it. And the results were astounding. Here were patients with metastatic melanoma, previously extremely resistant cancer, scattered all over the body like this, with dramatic responses after about 4 months of therapy. You can see the change. It's in much improved health. It took just 9 years from the discovery of the gene to get that drug into the clinic and to cause these dramatic changes, which is a very fast development program. But it took 9 weeks for the cancer to develop strategies to overcome that and to drive resistance. So that's hugely challenging. That's what underlies that poor outcome from those subsequent targeted drugs. And as Bert Vogelstein said, resistance to targeted therapy is a fait accompli. The time to recurrence is simply the time taken for the subclone to repopulate the tumour. Tremendous challenge to the concept that treating late-stage cancer with targeted therapies will really deliver long-term benefits. And why is that? Well, it's about the fact that cancer is not only unique in an individual person, but it's also evolving. And that treatment with these therapies drives further evolution and drives the development of resistance. As shown with this paper by Charlie Swanton, and you can see the way from the initial cancer, you get massive deviation, looking rather like Darwin's trees. However, out there, what is being said? So this is from the front page of the MD Anderson Cancer Center website. So here's the myth of personalised medicine, that we'll take a bunch of people with cancer and we'll molecularly profile them, and we'll look at a variety of prognostic markers, and every one of them will be allocated, and every one of them will receive a special medicine. That's what personalised medicine in cancer, that's the promise, or the hype. Another way of doing it champions oncology. So they will take a bit of your cancer, they'll put it into a mouse, and they'll expand it, and they will test all the sorts of treatments, and they'll see what your treatment options are, and you can begin treatment, targeted treatment, as indicated. These are called avatars, or patient-derived xenografts, PDX. Their data is reproducible, and it is predictive, but it's slow. And actually the time taken to go through this is about the time taken for a patient to receive a therapy, for them to develop resistance, and for their tumour to be significantly different from when it was here. So by the time you get the answer, the cancer's moved on. And it's extremely expensive. So some see this as a great epitome of personalised medicine, but others say they're far from ready. Where's the reality? Well, there was pancreas cancer is one of the most challenging cancers out there. The survival is still only 5%. And people are working hard to try and develop new therapies. This was a trial of personalised medicine, the IMPACT trial in pancreas cancer. 93 people screened, 74 analysed, 22 eligible, no patients enrolled on the therapeutic trial. Why? Some of them had died or were not well enough. Others had to start standard therapy before the result was available. Some of them became ineligible. So there's a big implementation challenge here, which is very difficult. In my own trial, so this is what I'm trying to do. I'm trying to do this. I'm working with some colleagues here. We're taking patients with colorectal cancer, with metastatic disease. We're analysing their biomarkers. And we have a plan for 6 molecular cohorts and 1 non-stratified cohort for those who are not specifically allocated to a molecular cohort. The reality of this is that although it's open in 90 hospitals, we've registered 424, we've randomised 113, only 32 of those are actually into molecular cohorts. And as we sit here today, although these plans are all in place, in fact this cohort has already opened and closed. It didn't work, and we're getting a new drug to look at in this group. The only cohort we've got open at the moment is this B cohort, looking at that wonderful new drug, aspirin. This cohort is all in agreement and about to start. These ones are in negotiation with pharmaceutical companies. And I could wax lyrical and tell you about the challenges of doing this in real time, and that perhaps will be something we can come back to. So there are real challenges of implementing this in the clinic. What about non-malignant disease? I'm no expert, but here's one of the things that people talk about: cystic fibrosis, a single gene, a monogenic disorder, multiple different genetic variations, single gene affecting the transmembrane conductance regulator gene. Lots of activity in this area, wonderful promise. Precision medicine is saving lives. Here's an example, wonderful example of a chap aged 8, he's now 27, receiving this drug Calideco, a new treatment. Reality, Calideco is effective in treating 4% of CF patients who have a specific mutation, the G551D. So, we've got a long way to go. So the hype applies also to preventative precision. Welcome to you, 23andMe. This is what they're saying. Welcome to you. Learn more about yourself. See what your inherited risk factors are. So the truth of personalised medicine, what's the hype, what's the reality? Sequencing the human genome, Gleevec for CML, going up that hype curve, short remissions with many of these agents, understanding of tumour heterogeneity and clonal evolution. So where are you? Where are you on this hype curve in your different diseases, your genome hubs? Can we ensure the right patient gets the right treatment? We as scientists are very invested in this. We've committed, many of us, our life's work to it. What are we trying to do? Is this becoming almost the meaning of our lives? The truth is that benefits are going to be much less obvious in some situations, and questions arise. Decisions are more nuanced. We need to be thinking about personal values, and the communication of risk becomes much more significant. Is this rich world medicine? Some of the questions that we're thinking about today come out of the fact that it's not all going to be Gleevec land where everything's black and white. Just a brief thought about what actually causes most disease. We know it's a mixture of genetics and environment, and there are some monogenic disorders, which is where much of the examples come from. In cancer, CML is a great example of that, cystic fibrosis, Huntington's disease. And treatments are becoming available for many of these, for few of these in CF, or for none. The Huntington's disease gene has been known for 23 years. No therapies coming forward. Most diseases fit in this middle group, like melanoma, like colorectal cancer, like many diseases. And we come to the new big fairy godmother, big data, which is going to provide the answer. Well, big data is really useful. Here's an example of big data, looking at all of the mutation, the incidence of mutation across the whole range of cancer. This is big data. And there's something very interesting has come out of this in terms of hypermutation seeming to predict benefit from immune checkpoint inhibition, one of the new therapies. So this group of tumours up here with very high levels of mutation seem to be responding to this new class of drug. And look at this is melanoma again, lung cancer, a subgroup of colorectal cancer where they have a particular molecular genetic profile. Another area where there's sort of a group of things showing benefit, where there's DNA repair deficiency in a number of diseases coming out, where there's huge complexity of the DNA response, but is that going to be able to be distilled into a test which can differentiate responders from non-responders? So this all raises the question of big data, which requires data sharing, personal participation, and we're going to be thinking about that today. So personalised medicine, huge scientific challenge, a very interesting scientific challenge, and we have some very exciting tools to be addressing in this area, and there is a big enterprise, and I'm far from walking away from this area. But I think we've got to face the challenge that black and white predictors of outcome will probably be rare. and that black and white decisions like Gleevec will be the exception rather than the rule. And Josh has identified some of the questions we're going to look at. So that's where I'm coming from the cancer perspective. And I'm going to hand over to Sir John Took now for his perspective. Thank you.